Our next speaker is Sergio Rutella, Professor of Cancer Immunotherapy in the University of Nottingham-Trent. He will discuss how immune infiltration correlates with P53 mutational status in AML.
Hello, my name is Sergio Rutella. I work at Nottingham-Trent University in England. I wish to thank the meeting organizers for giving us the opportunity to present today on immune correlates of TP53 mutational status in acute myeloid leukemia. This is my disclosure slide, which relates to research support from microgenics, nanostring technologies, and cure oncology. My lab in Nottingham is currently working on the high-dimensional analysis of the AML immunological tumor microenvironment using targeted immune gene expression profiling on the Encounter nanostring platform, but also geomics, digital spatial profiling, to try and elucidate the topography of the immune infiltrate and how this may correlate with immunotherapy responses. We also have an interest in identifying the oncogenic pathways that may drive immunological infiltration in AML. And this includes, of course, TP53, which will be the main focus of my presentation today. Let me provide you with some background on AML. So chemotherapy remains the standard of care for most patients with AML, despite recent approvals of novel drugs. Primary refractory or chemotherapy-resistant disease, as defined by failure to achieve complete remission of CR, occurs in 10 to 40% of the patient population. And the 5-year overall survival from first relapse for patients with AML is only 20%. So the investigation of new molecularly targeted and immunomodulating agents remains a high priority for the field. And there is an urgent need for predictive biomarkers to help identify patients who are more likely to respond to cancer immunotherapy. And we have recently identified the tumor inflammation signature as a good predictor of response to flutituzumab immunotherapy in AML. So this slide summarizes our recent work that used immune gene expression profiling of 290 bone marrow samples from patients with chemotherapy-naive AML. This analysis identified 3 distinct gene expression profiles, namely an interferon dominant profile, including high expression of interferon gamma, PD-L1, PD-L2, inflammatory chemokines, and interferon downstream signaling molecules, an adaptive profile, including high expression of T-cell, B-cell, and natural killer cell markers, and a myeloid profile, including high abundance of neutrophils, macrophages, and other cells of the myeloid lineage. These scores were then used in aggregate to stratify patients with AML into an immune-infiltrated and immune-depleted subgroup, as shown in panel A in this slide. And patients in the immune-infiltrated subgroup had higher levels of interferon-stimulated genes, T-cell and cytotoxicity markers, molecules implicated in antigen processing presentation, as well as actionable immune checkpoints, such as PD-L1, BTLA, CTLA-4, and TIM3. And as shown by the ROC values in panel C, interferon-related RNA profiles improve our ability to predict resistance to chemotherapy, compared with the cytogenetic risk classifier, which is still the gold standard in the clinic. We also used, as I recently mentioned, a geomics digital spatial profiling to identify protein co-localization patterns with potential prognostic relevance in T-cell infiltrated AML. This highly multiplexed technology allowed us to quantify 31 immuno-oncology related proteins in 10 bone marrow FFPEs from patients with newly diagnosed chemotherapy-naive AML. As shown in panel A in this slide, we analyzed the 240 geometric regions of interest, ROIs, with varying degrees of T-cell infiltration. And we identified prognostic 7 protein signature, which is referred to as signature 3 in panel B, and includes cytotoxicity molecules such as granzyme B, FOXP3, PD-L1, as well as STAT3 and BCL2. When evaluated in silico, our protein signature was deregulated at the RNA level in approximately 1/4 of TCGA AML patients, who mostly clustered in the immune-infiltrated subgroup. As shown in the mutation enrichment plot in panel B, the regulation of signature 3 genes is significantly correlated with TP53 mutational status. And approximately 55% of the patients with TP53 abnormalities in TCGA AML had the missense mutation, as summarized in panel C. AML patients with the regulated levels of signature 3 genes tended to have adverse cytogenetic risk, as shown in panel A, and had significantly shorter relapse-free survival and overall survival, as suggested by the Kaplan-Meier estimates in panel B. So to summarize this first part of my talk and the preliminary work that we have done in this area, we identified an experimentally derived protein signature, which is associated with immune-infiltrated AML, with TP53 mutations, and ultimately with poor clinical outcomes. We therefore asked the question whether TP53 could be a new genetic driver of immune infiltration in AML. So let me provide you with a background of the status of TP53 mutations, specifically in AML. So these abnormalities are known to occur in 8 to 10% of de novo AML cases and are associated with chemotherapy resistance, with high risk of relapse, and with dismal prognosis even after hematopoietic stem cell transplantation. So the functional consequences of TP53 mutation inactivation on host immunological responses have been largely overlooked in AML. The TP53 mutants studied thus far in AML do not show any evidence of gain of function mechanisms. We therefore asked the question whether or not TP53 abnormalities may correlate with the composition and functional orientation of the tumor immunological microenvironment in AML. So to be able to answer this question, we initially downloaded RNA sequencing data from TCGA, again, a collection of 147 cases of non-promyelocytic AML, including 14 patients with TP53 mutations. We then computed immune cell type specific and biological activity gene signatures as predefined linear combinations of biologically relevant gene sets, which were then correlated with prognostic molecular lesions, including TP53 mutational status, nucleophosmin-1 and PM1 mutational status, FLT3-ITD mutational status, and CHIP-defining mutations, and ultimately with clinical outcomes. We also provide primary bone marrow samples from 42 treatment-naive patients with TP53 mutated AML and 22 treatment-naive patients with TP53 wild-type AML using the pan-cancer IO360 panel from NanoString technology. And finally, in the translational component of this study, we analyzed primary bone marrow samples from 35 patients with relapsed refractory AML who were treated with flotetuzumab. 11 patients in this cohort had either TP53 mutations or 17p abnormalities with genomic loss of TP53. So moving to the next slide, panel A here shows unsupervised hierarchical clustering of 44 immune gene signatures in 118 TCGA cases with non-promyelocytic AML and available information on prognostic molecular lesions, including 14 patients with TP53 mutations. 2 patients subgroups were clearly identified, namely patients with immune-infiltrated AML and patients with immune-depleted AML. Compared with TCGA AML cases with favorable risk and intermediate risk molecular lesions, all patients with TP53 mutations showed high levels of immune infiltration and had higher tumor inflammation signature scores, inflammatory chemokine scores, interferon gamma signaling scores, and lymphoid scores, as summarized in panel B. In particular, the expression of immune checkpoints PD-L1 and TIGIT, and also molecules associated with an immune-suppressed tumor microenvironment, such as the Treg transcription factor FOXP3, were significantly higher in TP53 mutated patients, as summarized in panel C. And in line with previous findings, the Kaplan-Meier estimates of survival for patients with TP53 mutated AML were significantly shorter compared with TCGA cases harboring other risk-defining molecular lesions. We next profiled pre-treatment bone marrow samples from 42 patients with TP53 mutated AML. As shown in panel A, most patients in this cohort had missense mutations of TP53, which are often associated with TP53 loss of function. Principal component analysis in panel B highlights that immune cell type specific scores and biological activity scores distinguished patients with TP53 mutated AML from individuals with TP53 wild type AML. As shown by unsupervised hierarchical clustering of immune gene signature scores in panel C, the frequency of TP53 mutated cases was 87%, 75%, and 8% respectively in patients with high, intermediate, and low levels of immune infiltration. And also in line with our in silico analysis of TCGA AML cases, immune checkpoints PD-L1, FOXP3, interferon and granzyme B, a molecule recently associated with features of T-cell senescence and T-cell exhaustion, were more highly expressed in patients with TP53 mutated AML compared with TP53 wild-type AML. We next analyzed the immune transcriptomic profile at the gene level, and we identified a set of 34 differentially expressed immune-related genes between patients with mutated and wild-type AML, as shown in panels A and panel B. We will refer to this set of differentially expressed genes as TP53 immune gene classifier. The TP53 immune signature genes have not been previously implicated in the TP53 signaling pathway, as shown in the Venn diagram, by the lack of any overlap with hallmark TP53 gene sets downloaded from the molecular signature database. The differentially expressed molecules exhibited enrichment of KEGG pathways related to cytokine receptor interactions, autoimmune inflammatory processes, and IL-17 mediated and TNF mediated signaling, as shown in panels A and B. We then assessed the differentially expressed genes in silico for potential prognostic value in TCGA AML cases. Abnormalities in the 19 immune genes that were overexpressed in patients with TP53 mutated AML, by default, RNA upregulation, gene amplification, deep deletion, missense mutations, significantly correlated with worse relapse-free survival and overall survival estimates, as shown by the Kaplan-Meier curves in panel C. By contrast, the 15 immune genes that were downregulated in patients with TP53 mutated AML carried no prognostic significance, and these data are not shown here today. So we moved to the translational element of this work, and we asked whether TP53 abnormalities may identify a subgroup of patients with AML that may benefit from immunotherapy with flotetuzumab, a CD123 times CD3 bispecific DART molecule for redirecting host T cells to AML in the CP-MGD 00601 clinical trial. So this slide summarizes the clinical characteristics of the 35 patients in the flotetuzumab cohort. Median age at time of study entry was 54 years. 57% of these patients had primary induction failure, as defined by failure to respond to 2 or more induction attempts, and 17% of them had early relapse of AML, occurring less than 6 months after the achievement of complete remission. Approximately 70% of the patients had an unfavorable cytogenetic risk based on the 2017 ELN classification. Response in this study was assessed in terms of anti-leukemic activity, ALA, which was defined as either complete remission with partial hematopoietic recovery, partial response, other benefit, more than 30% decrease in bone marrow blasts compared to baseline, and non-responders were defined as either treatment failure, stable disease, or progressive disease. baseline bone marrow samples for immune gene expression profiling were available in 9 out of 11 patients with TP53 mutations and or genomic loss of TP53. Among these, 7 patients showed intermediate to high immune infiltration, as highlighted by the heat map in panel A. Time on treatment and time to patient death and or censoring are summarized in panel B for patients with TP53 mutations and or 17p abnormalities, including 2 patients who proceeded to receive allogeneic hematopoietic stem cell transplantation. Overall, anti-leukemic activity was documented in 45.5%, 5 out of 11 evaluable patients with TP53 mutations and or 17p abnormalities. So the reduction of bone marrow blasts in 10 patients with TP53 abnormalities for whom a post-treatment bone marrow sample was available averaged 42%, as shown in panel A. In TP53 mutated patients with evidence of anti-leukemic activity from flotetuzumab, the tumor inflammation signature score, inflammatory chemokine score, Treg score, and interferon gamma signaling score were significantly higher at baseline compared with non-responders, as summarized in panel B. And again, highlighting the association between response to T-cell engagers and the T-cell inflamed tumor microenvironment. Median overall survival from study entry was 4 months for patients with TP53 mutated AML or 17p abnormalities, as shown in panel C. And this survival estimate compares favorably with survival predictions for TP53 mutated patients with primary induction failure, median overall survival of 1.16 months in large AML series, such as the HOVON cohort, as shown in panel D. And finally, further in silico analysis also suggested that median overall survival is not dissimilar between newly diagnosed HOVON cases with TP53 mutations, 3.58 months, and HOVON cases with wild-type TP53 and primary induction failure, 3.78 months, panel E. So in conclusion, immune transcriptomic analysis of in silico and wet lab cohorts of TP53 mutated AML suggests the presence of high T-cell infiltration and high expression of immune checkpoints and interferon gamma signaling molecules, compared with AML subgroups with other risk-defining molecular lesions. Experimentally derived TP53-related immune gene signatures are significant for survival prediction in AML. And finally, immunotherapy with flotetuzumab has shown efficacy in individuals with altered TP53 status, with an overall reduction of bone marrow blasts averaging 42%, and with evidence of anti-leukemic activity in 45.5% of the patients. And I wish to conclude my presentation by thanking co-authors and collaborators. And the special mention goes to my colleagues, Ian Davidson-Moncada at Microgenics and Sarah Church at Nanostring Technologies, for their fantastic support to all of this. And thank you very much for listening.
What is the recommended input of RNA for the analysis of bone marrow samples?
Yeah, I'm ready. So the recommended input of RNA for the analysis of bone marrow samples will be between 100 and 150 nanograms per ml. This is what we have used consistently with excellent results in terms of quality controls.
Did you use purified leukemia blasts in your experiments?
So we did not use purified AML blast in our study because the main objective of this was to get a comprehensive overview of the bone marrow infiltrate, which of course includes immune cells. So all of these studies were performed using unseparated bulk bone marrow preparations.
Is there any evidence from solid tumors that P53 mutations associated with response to immunotherapy?
I'm ready. So there is actually evidence that solid tumors with TP53 mutation may be amenable to respond to immunotherapy. There are very good studies published in patients with lung cancer showing that individuals with TP53 mutations have higher levels of immune checkpoints, and they tend to be more responsive to PD-1 blocking strategies.
Are other genetic drivers of cancer associated with a hot tumor microenvironment?
There is evidence that other genetic drivers may correlate with a T-cell inflamed or interferon gamma dominant immune microenvironment. This is evidence, again, published in the field of lung cancers. Patients with mutations in KRAS and NRAS tend to have higher levels of interferon-related molecules, and again, could be particularly responsive to immunotherapy. So this multi-cohort AML study used a variety of highly multiplexed technologies, including targeted immune gene expression profiling and protein profiling, to dissect the immunological landscape of AML. The study identified patients with immune-infiltrated AML and immune-depleted AML, and these immune landscapes were shown to be highly predictive of responses to current treatment, including chemotherapy and immunotherapy. And particularly patients with TP53 mutated AML tend to have very high levels of T-cell infiltration and high expression of actionable immune checkpoints, including PD-L1 and TIGIT, and also increased expression of interferon gamma signaling molecules, compared with AML subgroups with other molecular lesions. We identified an experimentally derived TP53-related immune gene signature, which we used in silico to determine whether these genes have any prognostic relevance, and this was the case. So patients with higher expression of TP53-related immune genes tend to have a worse prognosis when treated with conventional treatments, including chemotherapy. And finally, our study also wanted to explore whether immune profiles can be used to predict response to a novel immunotherapy agent named flotetuzumab, which targets CD123 on AML cells and CD3 on T cells. And our study in 35 patients with chemotherapy refractory AML showed that patients with increased levels of interferon gamma related molecules tend to respond to flotetuzumab.